finding transmission clusters. And this is now, again, a very epidemiology specific question that we're going to try to address using all of the analyses that we did earlier and kind of defining this as a computational problem. So first, I'm going to just define what are transmission clusters. So just a little bit of terminology and definitions. Then I'm going to talk about what are molecular clusters versus what are transmission clusters. And again, this is going to be kind of a definition-based discussion just to introduce you to these terms. And then I want to end with these two sections of how can we find these molecular clusters from sequences? And then also how can we find these molecular clusters from phylogeny? So kind of applying all of that evolutionary biology stuff that we've talked about these past few weeks, actually applying them to epidemiology. So first, what are transmission clusters? That's kind of the focus of today's lecture. So transmission clusters, so a transmission cluster is a group of infected persons who are connected by transmission. So it could either represent recent and ongoing, so it can represent recent and ongoing transmissions in a population. And basically, if we can identify transmission clusters, then those transmission clusters can help inform where exactly we should target prevention efforts. So for example, with COVID-19, like let's imagine that some areas are maybe like more heavily impacted by COVID-19. Maybe we can prioritize giving those places vaccines. That was kind of a big issue early on in the pandemic. Or like if masks are limited, maybe we can try to give free masks to people in demographics that like we think are correlated with big transmission clusters. So yeah, so if we can identify where those individual transmission clusters exist, we can allocate resources to try to like prevent the spread of the virus there. And I also want to introduce this term risk network. So a risk network is a group of persons who are infected or susceptible. So it's not only just infected people, but it's also people around the infected people who are susceptible to becoming infected. So you can think of a transmission cluster as being a subset of a risk network. So the risk network is like all of the people in this community who could become infected. Either they're already infected or they're susceptible, so they could become infected in the future. Versus a transmission cluster is just the folks who are actually infected. So it's a subset of the risk network, just the people who are infected out of that overall risk network. If that kind of makes sense. And basically, kind of one of the key things is if we can identify transmission clusters, we can help find the most susceptible uninfected persons. There's a question in the chat, does this have to do with the contact network talked about in week 1? Precisely. So you can think of the risk network as being the overall contact network, but of one specific community. And then the transmission cluster is kind of the subset of that contact network that is actually infected. And yeah, and if we can identify those transmission clusters, the neighbors of the people in the transmission cluster, so the susceptible folks who are connected to people in the transmission cluster, but who are not infected themselves, those are where we might want to target our interventions. Yeah, any other questions? Any questions about those terms? So transmission cluster, risk network. Feel free to type in the chat, feel free to unmute. And there's a question, these are all local terms. Like, do you mean like local in terms of a community? Yeah, exactly. Yeah, so these are all local terms. So you can have multiple transmission clusters within a city, even on campus, like on UCSD campus, each of those buildings where they were like, oh, we found COVID in the wastewater in these buildings, like each of those would be a transmission cluster, even within the same campus. Yeah, great question. Okay, so how do we actually identify transmission clusters? So one way that you could do it, and this was kind of like the historical way that they used to do it, was using viral case surveillance data. So in general, you would just kind of see, let's look for an increase in the number of diagnoses in a particular geographic area or population. So like maybe we have COVID cases by zip code, and we would see like, oh, this zip code is actually increasing really fast. So let's zoom in over there. Oh, there's a question in the chat. Does susceptible people include those who are not near the transmission cluster? but can easily be infected due to disease, age, etc. Well, so if you're not close to the transmission clusters, then you by definition can't be infected because you're not going to be exposed to the virus. So susceptible includes anyone who might become infected, which implies that they should be somehow connected to the transmission cluster. Yeah, great question. And with COVID, it's a little bit less obvious, but like for viruses like HIV, which is primarily through sexual contacts or through injection drug use, if you are not connected to like where it's spreading, or if you're not partaking in whatever actions that it could transmit across, even though technically you could hypothetically get HIV if you were exposed, because you're not doing stuff that exposes you, you wouldn't be considered susceptible. Yeah, great question. So yeah, so we could look at the viral case surveillance data to see what areas do we just report more and more viral cases occurring. And I really want to emphasize that, note that if you just notice that the cases are increasing, that doesn't necessarily automatically mean that more people are getting infected rapidly. Because remember, there's a distinction between cases and actual diagnostic tests, and people could be infected, but without having been tested and diagnosed. So like with COVID, that was especially a big deal, where like if you increase testing, by definition, you're going to increase the number of people that you detect as being infected. It's not that you created new infections by testing, it's just that those infections already existed, but you didn't see them before. So yeah, with viral case data, you have to be super, super careful that a growth in the cases that you're reporting doesn't necessarily mean a growth in the infected population. It could be growth due to increased testing. So you just have to kind of weigh those different factors. Okay, so that's one source, is just looking at the viral cases. Another source is partner services in contact investigation or contact tracing. So basically, what we could do is every time we detect someone who's been newly diagnosed, we can have a survey or an interview where we ask them, who have you interacted with recently? So for COVID, this does happen sometimes, especially like early on in the pandemic. And like this is very common with things like sexually transmitted infections and stuff like that, where if someone tests positive for like HIV or gonorrhea or something like that, you want to ask them like, hey, who are your recent partners, just so that we can get those folks tested as well. So you're not directly detecting people by going out and testing them, but people who already came in, you can gain that information by surveying the people that you did see. So this is really common for HIV and STIs, but it is used in COVID-19 as well. Okay, and then the last type of data that I'm going to talk about is molecular surveillance data. And this is really the kind of brand new thing that's been the recent development. And it's where you try to use molecular biological data, so for example, sequence data, to try to detect these connections. So you try to identify who might be connected in terms of transmission clusters purely based on some type of biological data that you collect. So in our cases, we're always going to be just using sequence data. So we're going to be only talking about genome sequence data from viral samples that we collect. And yeah, this is a relatively recent thing. Okay, so any questions about these, the different ways that we can try to gain information about identifying transmission clusters? Feel free to type in the chat, feel free to unmute. So there's a question, what exactly is molecular surveillance data? So it's any type of data that we collect from, like, so molecular biology, so either DNA sequences, RNA sequences, proteins, if we have that type of information, some type of molecular biology data from people who tested positive. Yeah. Cool, okay, so now I want to distinguish between molecular clusters versus transmission clusters. So what we talked about earlier right now was all about transmission clusters, and now I'm introducing a new term called molecular clusters. So a molecular cluster is a group of persons linked by molecular data. So that biological, like molecular surveillance data I just talked about, a molecular cluster is a group of people linked by that molecular data. And again, in our purposes, it's always going to mean sequence data. And what I want to kind of emphasize to separate these two terms out is that a molecular cluster is a subset of a transmission cluster, because not every single infected person gets their molecular data collected. So for example, I don't sequence every single person who tests positive for COVID. In an ideal world, we would, but in reality, we have the diagnostic positives, and then a subset of those people actually get sequenced. So yeah, so the molecular cluster only consists of people in the transmission cluster who got sequenced. So just to kind of, again, summarize the hierarchy, we have the risk network, who is every single person who's either infected or susceptible to being infected. Then a subset of that is the transmission cluster. And you can have multiple transmission clusters in the risk network. And then a subset of a transmission cluster is a molecular cluster. So out of the people in this transmission cluster, who have I actually sequenced? Does that kind of make sense? And then when we're doing molecular clustering, we have to really be conscious of missing data. And this is actually a big problem with molecular epidemiology. And if you ever end up researching this in the future, one thing I've noticed is that people kind of use these two terms interchangeably, and they're not the same thing. And I've noticed in very legitimate research studies, they do molecular clustering and then basically assume that they've now estimated the transmission cluster, but you have missing data in these molecular clusters because you didn't sequence everybody. So it's really important to be conscious of the people who were diagnosed, but were not sequenced. So the people that are in the transmission cluster, but they're not in the molecular cluster. And you also have to be really conscious of the people who were not even diagnosed at all. So people who are in the risk network, but not in the transmission cluster. So yeah, missing data is really important to be aware of. Okay, any questions about this? The distinction between molecular versus transmission clusters? Anything like that? So what does being conscious look like? That's a good question. So being conscious could be it'll become more obvious when I talk about some of the techniques that are used for finding molecular clusters. But basically, if you make estimates about all of these people are from completely different transmission clusters, for example. Let's say you do transmission, let's say you do molecular clustering and you come up with these small groups and you say, oh, each of these people is in a different transmission cluster. But it could be that there was one person that you were missing that could be a missing link between all of them. So you sometimes have to be conscious of what if there was someone that I could have seen, but I just happened to not see, that if I did see them, it would connect all these people, if that kind of makes sense. And there's a question, I thought diagnosis equals being sequenced. Not quite. So when you do the nasal swab and they do the RT-PCR diagnostic test, on campus, if you ever had to do one of the vending machine COVID tests, those ones do not generate actual sequences. It basically just kind of lights up if there was COVID detected, or it doesn't light up if there was no COVID detected. Versus sequencing is actually a little bit more expensive. So from sequencing, I can diagnose, but not all diagnosis is sequencing based. Yeah, great question. Yeah, any other questions about this stuff? Molecular clusters, transmission clusters, diagnostic tests, anything like that? So how does one discover a missing link? You just keep looking. Yeah, there's no real good way. So what you usually want to do is come up with some simulation-based subsampling. So if you can kind of see, with simulation data, you can kind of simulate what actually did happen. So then you can then subsample your simulated data and then see, based on my subsamplings of my real simulated data, what does the inference kind of impact? So if you can see you might be able to make confidence interval types of estimates of how confident am I that these results would occur versus would be different as a function of subsampling. So maybe even if you subsampled like everything, the result wouldn't change. You can kind of explore those types of questions with simulations. There's not really like a one size fits all type thing. Nice. Okay, so how do we actually find these molecular clusters? And again, molecular for the context of this class just means sequence data, so viral genome sequences. So imagine that I collect a viral genome sequence from person X and person Y. So these are two people that tested positive for COVID, for example. And then I do the experiment where I actually kind of collect the nasal swab. I spend the money to sequence it. Now I have a genome sequence from this person, a genome sequence from this person, using all those techniques we talked about the past few weeks. So if X and Y were infected by the same exact person, so there's another person who infected person X and infected person Y, the pairwise distance, so this dx of y I'm saying is the pairwise distance between their genome sequences. So the pairwise distance between the genome sequence of X and Y will be super small. So in other words, the viral genomes are going to be super similar. And that's because not enough time has passed, so there's not going to be very many mutations between their two viral samples. Versus if X and Y were not infected by the same person, but they're in the same group, like they're in the same transmission cluster, D of XY probably isn't going to be as small as if they were infected by the same person, but it's going to be pretty small. There's a question, what is pairwise mass? So this is pairwise distance. So like if I basically take their two genomes and I count the number of letters that are different between them. So just count how many mutations exist that are different between these two genomes. And that's what I'm going to be calling the pairwise distance. Yeah, good question. So yeah, this is just saying like think of D of XY as the number of mutations between these two genomes. Like how many letters are they different? So yeah, so if X and Y were in the same transmission cluster, but they were not necessarily infected by the same person, this pairwise distance, the mutations that are different, it's going to be pretty small. It's not going to be as small as if they were infected by the same person, but it's going to be pretty small. Versus if X and Y were just completely unrelated, like they just got infected in totally different places, the mutation difference, the pairwise distance between their sequences is going to be pretty big. So the general thought process here is, what if I place a link between person X and person Y if their pairwise distance is very small? So how would this turn into a computational problem? We can think of like, well, what if I take every single pair of individuals, so like person X and person Y, person X and person Z, person Y and person Z, just take every possible pair of people, and then let me compute the pairwise distance between their viral genome sequences. And then if that distance is small, let me just put a link between them. Let me connect them together. And then each resulting chain of links, I will define as a molecular cluster. And just real quick, I want to show an example because I think this is a little bit confusing the way that I worded it. But let's pretend that we have these four people, A, B, C, and D. And then on these gray dashed lines, I've drawn what is the pairwise distance between each of their genomes. And I'm doing it as a proportion just so that it's like a percentage. So you can think like, oh, they're 1% different, the genome sequences. This one is 5% different. This one is 2% different. So I'm just drawing these as percentages of how different their genomes are of the viruses that you collected. So what I could do is like, let's pretend that I've decided 0.01 is a good threshold for determining small enough. So let's say that somehow I did a bunch of like maybe through surveys or like somehow I've worked with public health officials and we've come up with the idea that for this specific context, 0.01 is small enough that we consider them to be linked. What I could do is just connect every pair of individuals whose pairwise distance is less than or equal to 0.01. So here, the pairwise distance between A and B and the pairwise distance between B and C, those are the only ones that are less than or equal to 0.01. And then these all were not connected. That's why they stayed as gray dashed lines. They were too big. And then the chains in the resulting network. So I basically just look at the resulting chains that I get, and I'm just going to say those are my molecular clusters. So I'm going to say person A, B, and C are in one molecular cluster, and person D is in a different molecular cluster. Does the idea kind of make sense-ish? And kind of going back to Kevin's question earlier of like, how do we account for if people are missing? Imagine if person B just never existed. If person B never existed, A and C would no longer end up in the same molecular cluster. So that's kind of what I mean by like, you got to be extra careful, just being wary that you might have missing data. So like maybe there actually exists a person E somewhere that could connect person C and D or something like that. So you just got to be really, really critical of the results that you get. There's a question, why is 5% too high? So I just kind of made up the example. So just somehow you determine that 1% is like the big enough threshold that like anything less than that is that they're small enough to be linked. Anything bigger than that is big enough to be different. Yeah. And in practice, like those, it's usually that they figure out these thresholds by looking at survey data. So what they do is, if you know which people actually were infected by each other, so like let's say that you do contact tracing and you can overlay information about like, oh yeah, all of these people got infected at the exact same time, at the exact same wedding, and then these people did not get infected at the same location. You can try to use that type of information to see what threshold will make it so that your molecular clusters, like the people you group together, lines up the most with people who you know based on the contact tracing actually were together. So you kind of will try to inform that by doing some type of surveys or some type of contact tracing or something like that. And then again, just like most things that we do in this class, fortunately for us, we don't have to implement this from scratch. So there's a tool called HIV Trace that was originally made for HIV, but it's been used for other viruses, including COVID-19. And yeah, this is kind of a common tool that people use to do precisely this approach that we talked about, where you compute all the pairwise distances, define some threshold that you think is appropriate, and then it'll link together people that are in the same, like within that pairwise distance. And there's a link to the GitHub repo on the slides if you want to kind of take a look at their code base. Okay, so that was how we infer molecular clusters from sequences. Any questions about that before I move on? And I really want to emphasize that like decision of what threshold is appropriate is like super hand wavy. So that's another thing that if you ever do this type of research in practice, you want to be really, really critical of like, how do my results change if I change that threshold? So usually in practice, what they'll do is like, if you say 1% seems like the right number, maybe you'll do like 0.5%, 2%. You'll maybe do like a range of thresholds around it. Take a look at how the results change, and then hopefully whatever public health takeaway that you have should look similar no matter what. If it changes significantly, then that means that your results are not very stable. Cool. So any questions about that? Any concerns? So that's generally how people find molecular clusters from sequences. You compute the pairwise distances, and then if those pairwise distances are below some threshold that you've determined is appropriate, link those people together. Okay, so how can we find molecular clusters from phylogenies? Because remember, we did all of that hard work to infer the phylogeny and for the evolutionary tree of these viral sequences. So it would be nice if we could use all of that hard work that we did to hopefully improve the results of those clusterings. So just to refresh your memory, how is the transmission network related to the viral phylogeny? Imagine you have some transmission network. So nodes are individuals. These edges, these links represent transmission events. And if we zoom in on one of these transmission events, and this is kind of a review from the first week. If we zoom in on one of the transmission events, you have a bunch of viral lineages just evolving within the host. And then at the time of transmission, one of those lineages, so one of the leaves of this evolutionary tree, is randomly picked and then goes from the host into the recipient. And then as time progresses after that, both the evolutionary history in the host and in the new recipient continues to evolve. And then we can imagine that if both of these people come in for sequencing, the sequencing process is equivalent to me just kind of picking a couple lineages, a couple of these leaves from both of these people, and then I'm actually observing this tree here, just the pruned version of it that only has the leaves that I was actually able to collect with that nasal swab. All right, so hopefully people remember that from a couple weeks ago. So the question is, let's assume that using all of those fancy schmancy phylogenetic inference tools that I talked about a couple weeks ago, let's imagine that I use those tools and I'm actually able to accurately get this evolutionary tree. How can I do molecular clustering from that tree directly? So the viral phylogeny is heavily constrained by the transmission network, right? Each of those branching events can only happen at the time of those transmission events. So the transmission network places a bunch of constraints on what the evolutionary history is able to even look like. So if you imagine we have this big evolutionary tree, and let's say person X and person Y, the viral genomes we sampled for them are somewhere on this big tree. So if X and Y were both infected from the same person, I would expect that X and Y are going to be really, really close together on that evolutionary tree. So like if this is the whole evolutionary tree, X and Y are probably going to be like right next to each other. Versus if X and Y were not infected from the same exact person, but they were infected kind of in the same group. So they have this, they're part of the same transmission cluster. X and Y might not be right next to each other, but they're probably going to be like pretty close. Like maybe there'll be a couple branches off in the tree. It's not going to be on like opposite ends though. They're going to be still pretty close together. Versus if X and Y were infected from completely unrelated sources, we would expect X and Y are just going to kind of be super, super far apart on the tree. They might even be on like opposite sides of the tree. So they're going to be super, super far apart. So then this pattern motivates another idea. This pattern kind of motivates the idea that we can theoretically define these molecular clusters using relationships within the phylogeny. So using relationships within the evolutionary tree. And I kind of want to emphasize there's a bunch of different ways that we can define clusters in a phylogeny. So for example, you can imagine, let me find a subtree such that the maximum pairwise distance between any of the two sequences under this point is, below some threshold. So like maybe you want to separate them out as far as you can such that like all of the samples are in the same subtree and none of them are more than some threshold distance apart. Or like maybe you want to cut the tree in some way. So maybe like you want to, I don't know, like minimize some distance or somehow you want to cut the tree and then the resulting kind of pieces of the tree each define a molecular cluster. So there's a bunch of different ways that on a given tree, you could define clusters. And then also you could do something similar to HIV trace, but instead of using pairwise distances of sequences, you can use pairwise distances across the tree. There's a question, how far is close or far on the molecular tree? So just like with HIV trace, where we have to tell it some threshold, same thing with here. We would have to have some metric that we define of close versus far based on survey data. So we could theoretically maybe look at a bunch of people who via contact tracing, we know these people were all from the same event, and then look at how far apart they're, look at the relationship between their sequences on the whole overall tree. So yeah, we don't necessarily know it in advance. We have to figure it out from other types of kind of like contact tracing stuff. Yeah, so there's a bunch of ways we can define clusters. I don't want to get too in-depth on any specific approach because you could do all sorts of objective functions on the tree. You can kind of think of the structure of the tree as being super flexible and just cutting it in different ways is going to end up with different types of clusters. But if you're curious about it, this is a tool that I created that we actually wrote a nice paper about. Feel free to check out the tool, but it basically, actually, maybe I'll just pull up the GitHub repo. Basically, we have a bunch of different ways that you could define clusters, and you just kind of pick which one you think is appropriate. You pick what threshold you think is appropriate, and it'll do it super, super fast. And if you're kind of curious about the theory behind this tool, you can look up the paper. The paper is linked to on the GitHub repo. Let's go away. But yeah, it's like a pretty mathy paper, but we end up applying this tool to viruses. We apply this tool to like bacteria and like microbiome research. We define a bunch of these algorithms, stuff like that. So yeah, feel free to check this out. It's a little bit advanced, so you might want to wait till like if you ever take CSE 100. But if you want, you can also check out the code base, because I don't think the code base is too crazy. It's literally just one file. I like writing simple code. It's kind of like all in here. Cool. Okay, so yeah, so the key takeaway here is basically, so that, I mean, that's everything that I have for today, but basically the key takeaway here is we've learned about how we can generate these sequences. We've learned about how we can infer evolutionary trees from these sequences. And now here's a bunch of different ways that we can try to separate out people into these kind of risk groups to determine what groups of people are kind of most at risk for our intervention. And there's a bunch of different ways to do this. And I've given you 2 tools that commonly are used to do this.